When I was a first-year graduate student, I only had a hazy idea of what I wanted to research. I knew I liked material science, transport phenomena, and interesting math problems, but beyond that, not so much. I eventually came across an intriguing class of materials composed of polymers, proteins, macromolecules, and colloidal particles that interacted in complex ways that collectively gave rise to unique physical properties. Colloidal particles sedimenting under gravity can undergo an instability, rupture, and just cause the entire material to fail. Motile bacterial cells can interact with their neighbors and form intriguing patterns as they glide and spread along a petri dish. Subcellular molecular motors can drive filaments to drive and glide along an interface, causing unique patterns like a rotating tornado. I found it fascinating that if I were to take an individual component from a complex material, a single agent in isolation, this system was very easy to understand. But the moment I take that same easy agent, take it into a complex material and allow it to interact with its neighbors, that same agent that was very easy to understand became impossible to understand. When I was a first-year PhD student, I joined a wonderful theory lab that set out to address these types of systems. And as I learned more about the field, I realized that our theoretical understanding of complex materials was very primitive. And in fact, today, it's still very primitive. So let's suppose we have a complex material like this, a colloidal gel. Here's what I might have done as a theorist trying to understand complex materials. This material seems a little bit too challenging to understand. They're interacting in weird ways. They're percolating across the network. Oh, I know. I'm going to first get rid of that particle because that particle clearly isn't interacting with its neighbors. It's all the way out in the bulk, but it's still a little bit too hard. I'm going to get rid of that particle, that particle, that particle, that particle. I'm going to get rid of all the particles, but hey, I still have two left. And maybe, just maybe, if I solve this problem, I'll understand the whole material. And I know what, this problem is just two atomically smooth spheres in an unbounded, incompressible Newtonian solvent. Words that are like music to a theorist's ears. So then I go over to these textbooks that I used to call the Bible. And these Bibles are written 30, 40 years ago. And these textbooks had solutions to this two-body problem. So when I was a first-year PhD student, the following thoughts entered my mind as I opened these textbooks. Clearly, I should be able to solve this problem. Okay, so there are some parameters in here like the distance between the two particles, the aspect ratio, no big deal. I'm pretty good at math. Let's turn the page. Okay, whoa, there's a lot of terms. Okay, log R, 1 over R is no big deal. I can handle this. Wait, no, 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 no! When I was a first-year PhD student, I was devastated. I was devastated that I had to learn all this math to keep up with the community, and even more devastated that even after learning all this math, the moment I take this system and slightly elongate the particles, goodbye Bible. The moment they start to swim, the moment they add a third particle and have many-body interactions, goodbye tables, goodbye everything I learned. Let alone something as complicated as this, I'm never going to understand a complex material and theoretically understand what's happening to a system like this. So as a first-year PhD student, I realized that understanding complex materials is a very daunting endeavor. And while bottom-up, reductionist, simple approaches like these are very valuable, it alone is not enough. It needs to be coupled with a top-down approach that embraces the complexities that are inherent in complex materials. So today, I'll tell you about two examples of a bottom-up and a top-down approach that are conducted in our lab. Let's begin with this bottom-up approach so that you get a philosophy of the types of problems we address to understand complex materials. To introduce this bottom-up approach, I'm going to introduce a very simple experiment conducted by a postdoc Daeyoung Kim in our lab and Sachet, who's a theorist in our lab, to understand this problem. So we take an optical laser tweezer, which uses a focused laser beam to confine colloidal particles to a single point in space. I'm going to take 2 of those traps and separate them by some distance r, and take 2 colloidal particles, trap them at these positions. And these particles are elevated into the bulk of the solution, far away from any boundaries, far away from any interface. I'm going to take the outer particle and translate the trap position at some velocity u1, and I'm going to take the center particle and hold it fixed there. The unique thing about the optical laser tweezers is that it generates a net external force, but no torque. Therefore, the particles are free to rotate if there's a torque in the surrounding solution. Therefore, as I rotate the outside particle, it's going to generate a fluid velocity in the surrounding fluid due to the no-slip boundary condition. And the fluid velocity will penetrate to the center particle. And because the external trap generates no torque, the particle is free to rotate. And we should, in theory, see the center particle rotate. So here is an experimental realization of this setup, where we have an outer particle rotating at some fixed velocity. This is a movie in real time. These particles are 5 microns in diameter, and you can clearly see that as the outer particle rotates and rotates, we see that the center particle has an angular velocity, and it's rotating counterclockwise. And this type of interaction is called hydrodynamic interaction. It's mediated by the fluid. You might have learned about pair potentials in your thermodynamics or statistical mechanics classes. Those pair potentials are conservative pair potentials. Here, these are interactions due to a non-equilibrium fluid dynamical effect. They're non-conservative. So those interactions are quite different. This one's mediated by the fluid. Good. So everything I'll tell you about today are conducted in low Reynolds numbers in the Stokes flow regime, where linearity, time reversible symmetry all apply. Now I can conduct the exact same experiment and vary the distance between the 2 particles from closest approach of 6 micron separation all the way to 20 micron and beyond. What you can clearly see is that at the closest separation, the center particle is rotating very, very, very quickly. And as I increase the separation distance between them more and more, the center particle over here is rotating very slowly. So now I can plot the angular velocity of these particles, the function of the distance between the pair separation. And you can see that the angular velocity of the center particle goes down as a function of distance, which is what you would expect, because for Stokes flow, whenever you have a velocity disturbance, those decay like 1 over r. So the further away you are, the less stress that's generated away from that point. So now this was very exciting to us and our experimental setup, because this is not so much of a victory of the theory, but more of a victory of the experimental setup. Because the symbols that I'm showing you here are data directly from the experiments, and the 2 curves, the yellow and the white curves, are theoretical results coming from the near field and far field theories from the Bibles that I showed you earlier. So as I said, we were very excited about this because we had not seen an experimental realization of fluid-mediated interactions to this degree of accuracy. And the reason why this works so well in our case is that we are looking at rotations and torques, not positions and velocities and forces. Positions are harder to understand in our problem because there's an optical laser tweezer that generates an external force. There's a harmonic trap, and the particles are located at that position. However, there's no net torque on it, so any epsilon degree of fluid disturbance is felt in angular space by a rotation of the particle because there's no external torque on it. That's why the sensitivity of the signal is so good in our experiments. Now we can of course extend this experiment to many more particles. Here I have a case of 3 particles. We have an external particle rotating, and you can clearly see that the center particles rotate very quickly when the outer particle is close to it, but when it's far away, it moves more slowly. And sometimes it can actually reverse because the outer particle will cause a lubrication effect that then twists the particle in the other direction. Good. So we can actually do this experiment with even up to 100 particles and look at scattering and many-body interactions between many particles within a fluid in an unbounded system. Good. So far, I've shown you a system where I can impose an external velocity on the outer particle, and as an output, we can calculate the angular velocity of the center particle, or in other words, the period of rotation. Well, we can actually go in the opposite direction. We can put as inputs a specified period of rotation, and also the trajectory doesn't have to be circular donuts. We can specify any arbitrary trajectory on it and specify a period, and we could do a little bit of fluid mechanical calculations in theory to spit out what is the output velocity of that outer particle along a given input trajectory and given input period time. So we can do that. Let me give you an example. Suppose we have a Cassini oval, which has this trajectory, and I specify a time of 15-second period of rotation. Okay? We can generate the calculation and spit out what is the velocity that the outer particle must take along this Cassini oval. It looks something like this. Isn't it cute? I love watching this movie. So the outer particle moves very quickly when it's along the semi-major axis and moves more slowly when it's along the semi-minor axis, because that's what it takes for the center particle to rotate at a constant angular velocity and rotate once every 15 seconds. So it turns out we've tried many different shapes and trajectories. They all exactly nail the period of oscillation and the trajectory that we specify as input constraints. So basically what we have is a pretty accurate clock based upon fluid mechanical interactions, which is kind of cool. Let's see, how much time do I have left? Let me see. Oh, I can calculate it. I think about 400 cycles left. Let's go. Okay, so far, I've done this problem for an incompressible Newtonian solvent as a validation of the experimental assay, and we have shown that this data and the signal is very sensitive and accurate and reproducible. Now let's wave goodbye to incompressible Newtonian solvent and say welcome to viscoelastic fluids, because the same experimental setup can be done for any arbitrary fluid. And the example that I'm going to show you right now is a surfactant solution, shown here, which can self-assemble into worm-like micelles that are nanometers wide and hundreds of nanometers or microns long in length. Therefore, they're very thin and long worms, and at the concentration of surfactant and salts that we add to our system, they can also form worm-like entangled micelles. And these worm-like micelles are extremely interesting and different from chemical polymer entangled networks because these worms are extremely floppy and soft and fragile. These worms can undergo KT level fluctuation and undergo scission, unlike many chemical polymers, but they can also do things like reptation and entanglement, which are more common with normal polymers. So there's a time scale associated with scission, reptation, and entanglement, and we can measure that by doing classical macroscopic rheology, which we've done right here. So you can measure the elastic and viscous moduli, and the crossover point between these two curves is called the Maxwell relaxation time. It gives you the time scale of the network reorganization due to scission, entanglement, and reptation. For this particular material, that time scale is about 0.25 seconds. So every 0.25 seconds, the network reorganizes itself. Good. So I'm going to repeat the same experiment, going back to the circular donuts, in this viscoelastic fluid. What's going to happen? Let's take a look. So what I'm going to do is to perform what's called a step strain and creep experiment. The outer particle is going to rotate. This red dot in the center particle right here indicates the angle, which I'm plotting here on the graph on the left as a function of time. The angle goes from 0 to 2 pi, and it loops on itself. So let's watch that movie again. So the outer particle is going to start rotating. You can see that the angle grows. The derivative of this slope gives you the angular velocity at steady state. And at 15 seconds, I'm going to stop right there and look at the creep of the center particle. Let's watch it one more time. Before the outer particle moves, you can just see rotational Brownian motion. Once it starts moving, it starts rotating, and interesting things happen when you stop. So what you can see is that it was rotating counterclockwise at steady state, but after the creep, it reverses clockwise. So let's take a look at the creep part of this experiment, because this is a very interesting thing. If I were to plot the angle from the time where it stops, it starts to creep as a function of time, we see that all the angles reverse in direction. So it kind of spins in the opposite direction of when it was in the steady strain part of the experiment. And they all decay for different experimental bead-to-bead realizations. They all decay. And compare that with the Newtonian solvent control, which is shown in these dots. So you can see across all of these Newtonian cases, as soon as the outer particle stops, it's a step function, and immediately the center particle stops rotating, and it just fluctuates due to rotational Brownian motion. That's why there's a spread. And in fact, this spread gives us exactly the Stokes-Einstein-Sutherland rotational Brownian diffusion coefficient. It's spot on. Good, so we're still figuring out the mechanism behind this relaxation and reversal process, but one hypothesis is something similar to these mechanisms, where it's a toy car where you can store elastic energy as you rewind in one direction, and as it releases that elastic potential energy that's stored, it's going to then reverse in the opposite direction, just like so. Again, we're still working out the mechanism, but this is an interesting hypothesis that we're working out right now. Because the viscoelastic network in the surrounding worm-like micellar network is generating strains and elastic, polymeric energy. And the moment we build up that elastic energy, and the moment we stop it, there's going to be relaxation of all the polymers that are now fully stretched out. So hopefully we can work out this mechanism soon. One interesting property about the data that we see here is that the relaxation is happening over the timescale of several seconds, or a second's timescale. But earlier I told you that the maximum relaxation time is 0.25 seconds. So we're finding that in this experiment, the relaxation is occurring 10 times larger than the intrinsic timescale of the Maxwell fluid. Our hypothesis for this discrepancy of the big timescale difference is the fact that in the highly wound situation, the fluid is very, very far away from equilibrium, all the way out in nonlinear land, where there are additional timescales in the worm-like polymer itself that's not accounted for in the Maxwell fluid description of the polymer, which is only valid for small deformations of the fluid away from equilibrium. So it's the linear response regime that the fluid behaves as a Maxwell fluid with a single relaxation time. But when it's wound many, 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 many times, who knows what the fluid is like all the way out in nonlinear land. Good. So, as one indication of this, we could actually take the same micellar solution, view it through a polarized filter, and what we're doing here is just taking a power drill that you can get at Home Depot and just start turning the motor. And what you're going to see is a polarized intensity of regions around the rotating element is shining very brightly, which means that there are large elastic stresses that are built up there, and the polymers and the worms are aligning locally there. That's why you see polarization. So presumably, in our experiments, which we're conducting right now through a polarized film, we're going to see large elastic elements stored in polarization forming between these two particles, which is indicative of the microstructure of the worm-like polymers present here. So it'd be really cool to couple the fluid mechanics with the microstructure of the worms locally between the particles. And this gets to an important point about the beauty of a bottom-up experiment. Because this is a clean, reductionist approach that gives a very accurate and reproducible measurement, we can really trust these measurements to provide some insights back into the theory land. So this is the Stokes equations coupled to an evolution equation for the stress tensor. And nonlinear terms can appear out here in this F term. But we can use this experimental measurement in arbitrary fluids to understand two-body interactions between particles, which actually form the basis of a large-scale simulation just like this. So the major advantage, or one major advantage of a bottom-up experiment is that you can use the data to inform theory and new simulation development. And it would be really cool to be able to have a million particle simulation just like this in arbitrary viscoelastic fluids. Such a simulation is not around yet, and it would be really cool if our experimental measurements can inform a simulation like this. And we're working on this right now inside of our research lab. A small variation of the experimental setup that I just showed you can be achieved by taking two optical tweezers very close to each other, which will then form an energy landscape as a function of distance that looks like a W function. And so now there's two local minima, and we can look at the hopping of this colloidal particle between these two local minima. And you can plot the position of that particle as a function of time, and it'll look something like this. It'll fluctuate, they'll hop, it'll stay in that new basin and hop back again. The distribution of these residence times for a normal incompressible Newtonian solvent will be a Poisson distribution. You can actually use the fluxes and the hopping between the two basins to tell you information about the transport properties and the depth of these barriers. We can also do something interesting, like make one of the traps deeper, so you can now make a tilted potential, and you can actually start doing some rare event like dynamics and study them in a viscoelastic solution. And in viscoelastic solutions, the hopping, the rare event escapes could all change in very interesting ways. In rare event physics in viscoelastic memory-containing fluids is, in my opinion, an understudied problem. Good. So the really cool aspect about these kinds of bottom-up simple experiments is it tells you something about the memory contained in the fluid, and you can use that data to then, again, go to large-scale simulations in viscoelastic complex fluids. That looks something like this. Okay, so hopefully that gives you a sense of the overall spirit and philosophy of our lab of using simplified, easy experimental setups to reveal something about deep theoretical and computational results coming out from the data. Now I'd like to transition over to our philosophy on the top-down approach of understanding complex materials, where we embrace the complexities that are present within these materials. And the example that I'll show you today of a complex material that we have studied in our lab is called active matter, which is any material composed of agents that are able to undergo self-motion. It could be subcellular molecular motors and filaments, it could be motile bacteria spreading along a petri dish, or you can actually make synthetic colloidal particles move through an asymmetric chemical reaction happening along the surface. Now, the basic theme of this community has been, let's take an active agent that can undergo self-motion, do something to it, like shine a laser, shine some sound waves to it, change the density, change the motility, the tumble rate, and see what happens to the pattern. See what happens to the distribution of those agents in space and time. In this project, I wanted to introduce you to the reverse problem, where I would like to specify a given distribution or a given pattern, and I want to know, what do I need to do to the original material to produce that distribution? So it's the inverse problem. So now this is a project that is in collaboration with Professor Jim Rawlings at UC Santa Barbara. Titus Kwa up there is co-advised between myself and Jim Rawlings, and Q-Hwan Choi is an experimentalist in our lab, making this all possible in an experimental setting. Good. So let's first understand the mathematical structure that some people use to understand active matter systems. And it takes a statistical mechanical description, a probability distribution of locating some agent at a position x, pointing in some angle theta at a given time t. Now, the evolution equation for this equation satisfies a convection diffusion equation that looks something like this. There's convection coming from the fact that the particles are swimming. There's diffusion in both position and angular space because there's thermal kT coming from the surrounding bath. Now, one could solve this problem But the angular degree of freedom right here is often annoying mathematically to handle. So what the people in the community sometimes do is to do an expansion, analogous to a Taylor expansion of this full distribution in angular space. Therefore, we have the 0th moment of the full distribution, which corresponds to the density. This just literally tells you where are the particles located as a function of position and time. The first moment below that is called the polar order. It tells you which direction are the agents pointing in as a function of position and time. One moment below that is called the pneumatic order. It tells you how are the agents aligned relative to their neighbors as a function of position and time. Now we can write an evolution equation for the density, and it'll also give you a convection diffusion equation that looks like this, except the convective term is now a function of one level down, the polar order field, which kind of makes sense because the density in space depends on where the agents are going. If it's moving in this direction, the density is going to be higher in this direction at the next time step. So it makes sense that the density is coupled to the polar order. Well, that means I need to write a separate equation for the polar order. And that equation is going to be coupled to the density and also one level down to the pneumatic order. So as you can see, we now get an infinitely coupled hierarchy of partial differential equations. Therefore, at the benefit of removing 1 degree of freedom of angle comes at the cost of having an infinitely coupled PDE. In practice, we can truncate this at some level and solve a closed set of equations. But there is a big experimental and practical limitation of this moments approach. So as a theorist, but also as an experimentalist, we often deal with images and data that look just like this. This is a microscopy image of fluorescent labeled bacteria along a chamber. All you see are bright dots, which are the bacteria positions in space, but they just appear as dots because the bacteria size is small compared to the pixel size of the camera. So we don't know where the bacteria are pointed or how they're aligned relative to their neighbors. Ain't nobody got time to calculate where that dot is oriented, which direction that dot is going. We have no angular information from experimental data that look like this in practice. Therefore, while in theory, this Taylor expansion and this approach is nice, in practice, oftentimes, it's not that helpful. So ideally, we would have a closed set of equation with just the density and doesn't depend on all these moments below, in the ideal case. Because then we would be able to write down a convection diffusion equation that looks like the following right here. And the convective term V is just going to be a function, a nonlinear function of the density, which is then indirectly coupled to all the lower order moments. And what I'm going to do is to impose this convection diffusion form, because this is the equation that I want, and figure out what the convective term is based on microscopy data. One can say, well, you can remove even more mathematical structure and go all the way to this form down here, which is time rate of change of the density plus some arbitrary function must be equal to 0. And we can just learn what that arbitrary function is. Fine. I'm going to call that the black box model because it contains the least amount of physics. The white box model contains the most amount of physics. We know exactly every term and how it's coupled to every moment. But the white box model, like I said, is not useful in practice. The black box model could violate fundamental laws of physics like mass conservation or negative densities. So what I'm going to show you today is the gray box and how we use this to understand these types of complex materials while embracing the complexities present in these complex materials. So in short, what we are doing is we're taking all the possibility of universe of what this function can be for the black box model and constraining it to a smaller space. And I'm a big believer in what we have achieved as humans through 400 years of physical knowledge. We shouldn't just dump everything and all the hard work we've done for 400 years of physical knowledge. We should use that. So we shouldn't underestimate human intelligence, HI. We're going to use HI to reduce this space to a smaller circle and then do AI within this small little space right here. So it's HI driven to a small space and do AI in that small little circle. That's what we're doing here morally in this gray box model. Good. So there's an important concept called time delay embedding, which is absolutely essential for these types of complex systems. So as I said earlier, a single snapshot of microscopy or an image can tell you information about the density because we know exactly where the agents are located at that particular time. So we get the density for free. But if I take one snapshot from there to there, from here to there, I take one snapshot, all of a sudden, I know where the agents are pointing. An individual agent moved from one spot to the next spot, so I actually know which direction they're pointing. All of a sudden, I know the polar order. I take another snapshot, all of a sudden, I know how the agents are oriented relative to their neighbors. And if I take a movie, I know all the moments all the time, and I can just take a sliding window all the time, and I know all the moments. So what's important about the gray box modeling is that this function that we figure out using the computer is as an input, we feed not only the current density, but all the past densities. And when you feed in all the past densities, it indirectly is feeding in information about these lower order moments. This is called time delay embedding. Now, to be fair, time delay embedding is not something we invented. It's been used in science and engineering for a long, long time. So let's suppose we take this famous example of a Lorentz attractor system, which is a minimal model that explains chaos. It's a coupled ODE that depends on three variables, x, y, and z. And it is a system that, like I said, exhibits chaos due to the fact that they're non-linearly coupled to each other. So the x position, the y position, the z positions are located as shown, and the lobe switching between the two butterfly wings are very difficult to predict because of chaos. Well, it turns out that as opposed to measuring x, y, and z, we can just measure the x coordinate, the current x, the one past x, and two past x positions, and just measure x only with no measurement of y and z, and recapitulate the salient feature of the Lorentz attractor system, like the butterfly manifold and the Lyapunov exponents that tells you about the divergence of the chaotic motion. And this is a very common experience as an experimentalist, and I'm sure you're probably experienced this too, is that you're working with a complex system and you're making some kind of measurement about that complex system. You probably only have a measurement of one aspect of that system, but not the full system state. Well, it turns out that there are cases where you can just take a measurement of that one measurable quantity in time and make a very good estimation of the unmeasured states in your system. Time delay embedding. This is called also observability. Now, in my discipline of chemical engineering, process system engineering is very common where time delay embedding is critical. There are situations where we know the temperature of a chemical reactor, but we don't know the flow rate downstream, we don't know the pressure downstream. Well, no problem. Let's just measure the temperature of a reactor as a function of time, get a history of that information, and we can estimate what the flow rate and the pressure is downstream based on embedding of the temperature data. So embedding is a very critical concept for understanding and modeling complex systems. Good. So let's return to this example of controlling active agents because that's what we like to do. Earlier, I told you that the system follows a convection diffusion equation, shown here, convection, diffusion. But let's add a couple of extra terms because we want to control them first. So let's add a controller, which might be able to induce a torque. So we're going to add a control convective term right there. And secondly, we want to be able to model complex interactions and many-body interactions between them. That's this term right here. This term is a term that kills everybody. Whether you're a biologist, chemist, physicist, it doesn't matter. Whenever you have many-body interactions between organisms, chemical components, quantum systems, condensed matter systems, anything, many-body interactions between components, organisms, chemical components, particles, always results in the fascinating world of the complexities of patterns and evolution and complex dynamics that we see in life, but it is also a term that's extremely difficult to understand because it is highly nonlinear. Now, one approach that people can do to take care of this very difficult term, and this is what I used to do for like the first 10, 12 years of my research career, is to take this equation and just sort of kind of like massage it, just work on it, massage it here and there. And ultimately what you'll get is you'll get a complicated integral with a collision kernel that will account for all these many-body interactions. And you need a closure relation for that collision kernel to actually make analytical and numerical progress. But that closure relation often only works for simple systems. And whenever you want to understand a complex system, that closure always falls short of my expectations. And even when I think that the closure relation is working and I'm getting the right results to explain a complex system, it usually is not. It usually is compensating for the 27 other terms that I'm not including in the equation. So even though I think I'm right for the wrong reasons. And at the end, I'm very dissatisfied that I haven't learned any physics at the end of it. So here I'm going to take an alternative approach, a orthogonal approach, based upon using the computer and letting it figure out the many-body interactions for us. So we're going to impose, like I said, a convection diffusion equation, and this convective term V is going to contain all the complexities of the system. It's going to include convective swimming. It's going to include the many-body interactions. It's going to include whatever the control torque is doing on my particles. Okay? So for people who are a little bit uncomfortable with this, of letting go of physical insight into this difficult convective term, this is why it's called the gray box model. One could put more structure in this convective term V based upon your domain knowledge in whatever complex system that you're modeling. If you want to go along the thermodynamic route and do the massaging of the equation, you can do that. And you can arrive at some collision kernel that then you put structure on that and you learn what that function is. In fact, people are already doing that. But for this particular application, we just want to control the agents, and I'm not going to put further structure in the convective term. So let me show you how we actually learn this function and use data to figure out what this equation is about. So we're going to do a simple Brownian dynamics simulation of self-propelled agents rotating due to thermal rotational diffusion and propelling due to a self-propulsive convective term. We're going to have a random collocation of torques, and here is the snapshot of the Brownian dynamics simulation movie. And for every random collocation of torque, you're going to see that the particles are reorganized and rearranged in space and time. And here is a plot of the density as a function of the position in one-dimensional space along Z. And the blue, the cyan data, are the raw data coming from the simulations. Now, what we're going to do is to use that raw data and learn what this convective term V is so that I'm arriving at a convection diffusion equation. Now, what's nice about this single convection diffusion equation is that it's a very easy equation that undergraduates can now solve. So you can solve this evolution equation, obtain the solution for the number density, and that's what's shown in the magenta right here, labeled as learned. You can see that when you train the data sufficiently number of times, the learned solution is spot on with the data that we're putting into our simulations. And if you're curious about what the learned function looks like, that's what's shown right here. So once again, this function, this arbitrary vector convective function, is learning the impact of swimming, many-body interactions, and the control actuator torque. It's all in this function right here. Good. So now that we have this function and this equation, we have basically used pictures from experimental and simulation data to understand what this convective function is. So that's where the cheesy title of my presentation here comes into play, picture perfect. We're using our transport and physics knowledge to put the basic mathematical structure into our problem and then using pictures to complete the equation. So to be clear, we have F equal ma dynamics, or experiment happening. We take pictures of that. Actually, maybe the young people don't know what this is. We can just take pictures of that system and then come up with a simple convection diffusion equation to understand the dynamics. And once we have this process of an experiment all the way to a gray box experimental simulation learned convection diffusion equation, we can actually do stuff with it, because now we have this simple convection diffusion equation that we could just solve. In our particular application, we are trying to control the density from one distribution to another distribution, for example, a set point. So in our particular case, we're going to take the gray box model, put it into a feedback controller, and that feedback controller will send data and signals back and forth in real time to the experiment or simulation to control the density and the patterns from one to another. I'm not going to get into so much of how the feedback controller works, but take my word for it, we're using a feedback controller called model predictive control, which is a very powerful feedback algorithm that calculates an optimal control problem over a finite horizon and repeats that calculation every time step and imposes the first time steps control policy at that time, and then marches forward using a model that we've constructed up here. So let's see an example of that controller in action. So we're going to return to our Brownian dynamics simulation. Now these have collisions, they're swimming, they have control. And we're going to try to do a user-specified control action on this simulation. So what we're going to do is to split the two peaks over here. We're going to get one peak to enrich by 75% compared to the left peak. We're going to juggle the peaks from right to left. So now the left peak has 75% more, and then go back to 50-50. Let's watch that one more time. And you can clearly see from the simulation data, the snapshots, the particles are interacting with each other. And here is the set point of the density on the left half from 0 to the left. The target is shown in the magenta dashed line, and the model predictive control is basically spot on with our user-specified targets of what we want the density to be on the left and right half. And finally, this is what the controller is doing to the simulation to make it all work according to our user-specified trajectory. Good. Okay, so now let's move away from simulation data and move over to experimental data. We're going to take a light-activated bacteria that has a proton pump along its membrane that can regulate the fluxes of proton based on light intensity. And as it regulates the flux of protons across the cytoplasm, it will then turn a flagellar motor in its bacterial body that causes it to self-propel. And actually, these types of bacteria have been used by a variety of researchers in very fascinatingly unique ways to form patterns based on light intensity. So here's some data on motility speed as a function of light intensity. And you can make the particles move and also tumble based on light intensity magnitude. So now, what Q-Hwan, student in my lab, is doing is taking these bacteria and putting a random collocation of light intensity, similar to what I just showed you with the simulation, but now a random light intensity, which will then cause the bacteria to form a particular pattern. Shown here is the bacterial density as a function of position, which is dynamically evolving in time. So what we are doing now is the exact same thing. We're using this as training data to figure out a convection diffusion equation of the bacterial motion as a function of position and time. And now this convective term V is modeling swimming. It's modeling many body interactions. They're rod-shaped, so their interaction is a little bit weird, right? Hard for a first principles analytical calculation to do. And who knows what the light controller is doing? There is a long cascade of biochemical interactions, biophysical dynamics happening of light then being converted over into actual physical mechanical motion. And all these three things are coupled to each other in likely nonlinear ways. And therefore, we're going to embrace the complexity and just allow the convection diffusion equation form to provide the structure that's needed for us to make progress on this problem, without worrying about the details of trying to rigorously work out each individual term, which, like I said, in my experience, have always been dissatisfying. When you try to do a first principles approach and try to do some kind of closure, I've been dissatisfied with that kind of approach. Good. So effectively what we've done is to use pictures and figured out the advective flux expression in our equation. And even more broadly, we're using pictures to come up with a conservation of mass expression. It's a picture-perfect equation based upon our experimental data. And once we have a model, we can do stuff with it, like cause the bacteria to undergo density waves along space. And Q1 took pictures of us, Titus and myself, from the lab website, inputted that, and you can make bacteria patterns and of faces, and you can make them morph from one to another in very weird dynamical ways. And the scale bar for these pictures are millimeters. So you can like almost see them with your own naked eye, the patterns form. So it's pretty cool to see them in real life. So what we're currently trying to do is make the active particles do stuff for us. So here is a really, one of my favorite videos of border collies herding sheep around. And it does it in a way that causes the sheep to move in a particular formation. So this is a movie of them playing Pong, the game of Pong. They have fluorescent LED lights. So just now the right-hand side team won. But it would be really cool if we can use materials and dope them with active agents with the right degree of control based upon knowledge of their equation of motion and equation of mass conservation to then do stuff to materials contained within soft matter. It just so happens that border collies are extremely good feedback controllers. So the summary of what I wanted to tell you about today is that pictures contain a lot of information. And when you combine pictures with transport knowledge, it's really hot. You can make a lot of progress, both from bottom up, but also embracing the complexity from the top down. I like to now back up and make an even bigger, ambitious project of what our lab is currently pursuing. Suppose we were to hand you a fluid containing anything. About 100 microliters of fluid containing polymers, liquid crystals, ionic species. It could be soap bubbles and films. Ideally, we would take that fluid and come up with an equation of motion. Once I have an equation of motion, we're set because we can solve complicated simulations, we can do material design, we can forecast material properties and phenomena. And so we would like to ideally go from some unknown fluid all the way to an equation of motion. Unfortunately, we don't know the relationship between the flux and the stuff, the conserved species. Traditionally, we have used rheology to provide the missing piece of information, which is how the stress tensor or the flux is a function of the gradients in velocity, the stuff. But there are many situations where macroscopic rheometer can't be used for a lot of the modern applications in science and engineering. For example, suppose you're working with a fluid that's extremely precious and rare, and you can't generate hundreds of milliliters of fluid to stick between a parallel plate macroscopic rheometer. Or maybe you're working with soap films and bubbles where all the dynamics are happening along interfaces, and stuffing it into a macroscopic rheometer isn't quite measuring the interfacial properties that you want to measure. Suppose you want to image the viscoelasticity of a biological cell membrane loaded with lipids and proteins and sugars connected to the actin cytoskeleton. We can get very good images, we can get nanometer resolution images, but we can't stick that into a rheometer. But what if we can use that really good nanometer resolution image to make some progress? What if we can take those images by taking really good pictures pictures of the phenomena or whatever fluid that you want to understand, use the right degree of transport knowledge based upon the relationship of the flux to the conserved stuff, and arrive at an equation of motion. Because in principle, this image should contain everything we need. It just requires the right amount of mathematical structure, the gray box structure, for us to make progress. Let me give you an example of what I'm talking about. So if I were to put in the Newtonian stress tensor, this would give you the Navier-Stokes equations, which is one of our favorite equations as engineers. Now we can solve this Navier-Stokes equations. In this case, we've solved it for a flow around a cylinder. It will look something like this. Okay? So now, suppose that this movie or these images were all you had. Just like an experiment. You're doing flow around a cylinder, this is all you have. And suppose that you didn't know that this was a solution that came from these equations. Let's get rid of that equation. This is all you have. The question is, can you use these images to recover the Navier-Stokes equations? Well, one approach, which has been conducted in the literature, is to say, I'm going to take my time rate of change and my velocity is some function of a candidate set of terms that is plausible based on mathematical and physical symmetries of vectors and tensors. Okay, so you can take the velocity itself, gradients of the velocity, and nonlinear combinations of those two. And you can come up with a set of candidate terms. And so this is kind of in the spirit of the gray box model in a way, in the sense that you're putting mathematical structure on the possibility of those terms contained in the equation. Well, it turns out that if you just use these images in a sparse regression with these terms, you'll recover exactly the turbulent term, v dot grad v, and also the diffusion term. And you'll recover the Navier-Stokes equations exactly, okay, with some sparsity term. Now, it's pretty clear that if you had more complicated fluids, like a non-Newtonian fluid that exhibits rod climbing, this may not work out so easily, because the flow around a cylinder may not sample all the possible configurations and all the terms that are actually present in a constitutive relation for a non-Newtonian fluid. It might be the case that we need to measure large normal stress differences in order for us to model the right kind of dynamics shown for the viscoelastic fluids. So a sudden contraction is known to measure large normal stress differences. So it might be the case that we need a secondary experiment. So there's a very interesting question of what are the minimal set of experiments in geometries that one would need to conduct for the fluid to sample all configurations of its constitutive relation. That's what we're thinking about right now within our lab. So the most broadest sense, what we are trying to do is use pictures to come up with a conservation equation. Now, we've learned in our undergraduate classes that the moment you have a relationship between the flux and the stuff, you're done. And we have plenty of examples of cases where the flux is just a negative gradient of the stuff. Fourier, Fick, Newton, they are all examples of constitutive relations that are negative gradient of stuff. But we know that fluxes are not necessarily linearly proportional to the gradient of stuff. Non-Newtonian fluids are a very common example. Many people modeling electrolyte transport in batteries often use a mass flux proportional to the linear gradient of an electrochemical potential. But that's likely not true in many cases where you have dense electrolytes. Who knows that they're linearly proportional to the electrochemical potential? Most likely not. It's probably nonlinear functions of gradients and their potentials. So there's a lot of insight that we can get from having the right constitutive equations and using images to provide information about the constitutive equation, I think is a very fruitful approach for science and engineering. It would be really cool for us to be able to look at granular materials, which are athermal solutions or athermal systems, so classical thermodynamics don't apply. But again, there are lots and lots of images of forces, mechanics, and stresses that I think we can leverage to come up with better models for constitutive relations for these types of granular materials. Same with larger scale geological systems like avalanches, mudslides, and glaciers, which are also classifications of granular materials. And we believe that there's a lot of information contained in the pictures and images to make some progress on coming up with the right conservation equations. In summary, we are a lab that believes that quantitative imaging and transport knowledge can make a lot of progress in our understanding of complex materials. If anyone is interested in finding out more, please contact our lab. Please contact any member of our lab anytime, and we are happy to chat with you. Thank you.